Hello, everyone. Welcome to BrainMap. We're so happy to see you all here in this beautiful spring day. So it is my great pleasure today to introduce our speaker, Dr. Jordan Farrell. So Dr. Farrell received his PhD from the Hotchkiss Brain Institute in Canada, where he discovered that a stroke-like event occurring after seizures is responsible for long-standing behavioral impairment, which won the Best Publication Award from the Canadian Institute of Health Research and Canadian League Against Epilepsy, and has directly led to two clinical trials. He then did his postdoc with Ivan Scholtes at Stanford University, where he received a K99 from NINDS to better understand how the hypothalamus controls hippocampal synchrony and epileptic seizures. He is currently an assistant professor of neurology at Harvard Medical School and a faculty member of the Rosamond Stone Zander Translational Neuroscience Center and FM Kirby Neurobiology Center at Boston Children's Hospital. His lab aims to understand the mechanisms by which heterogeneous cell types orchestrate synchronous network patterns for brain computations as fundamental as memory, which may be key to understanding the origins of pathological hypersynchrony in epilepsy. Using cutting-edge in vivo approaches to hone in on these mechanisms with precision, their goal is to restore circuit function with minimal side effects. As a step towards translation, they develop tools to uncover mechanisms of ultrasound neuromodulation. And as always, you will remember that BrainMap Seminar Series is co-sponsored by the P41 Funded Center for Mesoscale Mapping housed in Martinos. With that, it's my great pleasure to hand it over to Jordan. Thank you so much for being with us today.
All right. Thank you very much for the introduction. Really excited to be here. I opened my lab about a year and a half ago, so I'm still quite new to the area. But yeah, as Abbas mentioned, we're very much interested in the fundamental mechanisms of learning and memory and how synchronous brain patterns support some of those computations in the brain that are related to memory, for example, in the hippocampus, and how these actually might be prone to being hijacked in brain conditions that are associated with hyperexcitability, most notably epilepsy. So, okay, so I'll try and get rid of that. There. Otherwise, you're not going to see any titles on the slides. Okay, so usually when we think about hyperexcitable circuits, we think about epilepsy. Of course, it's very common. So at some point in your life, you've got about a 1 in 26 chance of being diagnosed with epilepsy, which is characterized by these unprovoked seizures. And really, a seizure is just excessive synchronous activity in the brain that produces some behavioral disturbance. And so, of course, hyperexcitability is not just something that's specific to epilepsy. We see the manifestation of hyperexcitability, for example, in Alzheimer's disease. So this is actually a study that was performed in mice, and this is EEG recordings. And you can see in a mouse model that has the pathological features of Alzheimer's disease, these highly synchronous discharges. They're shown as like these large spikes. And these can actually be blocked by anti-epileptic drugs. And then sure enough, when you test memory on this mouse model, if you give these drugs that actually block the seizure activity, it improves their memory dramatically. And even clinically, so if you record from somebody with Alzheimer's disease on EEG, very rarely do you see any sort of evidence of epilepsy. But in this study, this is actually done at MGH. You implant electrodes deep into the temporal lobe through the foramen ovale. You actually record all this seizure activity. So there's all these subclinical seizures going on. This person was put on the same anti-epileptic drug from this mouse model, and lo and behold, their memory improved dramatically. And the only time they really had like strong cognitive impairments was when they forgot to take their seizure medication. And then finally, just as one more example, we know that autism spectrum disorder is also associated with hyperexcitability. About 1 in 5 people with ASD also have epilepsy. And there's a high incidence of overlap in the genes that are known to be involved in autism that are also involved in epilepsy. And especially if you look in some of these conditions, you can see a very high abundance of epileptiform activity shown on EEG, even without a diagnosis of epilepsy. So these hyperexcitable circuits are often associated with these very synchronous discharges, which are likely to disrupt normal cognition. And so that's what we really want to understand. So how does excessive synchrony emerge in a hyperexcitable brain? And so the hypothesis that we're really trying to test in the lab is that the mechanisms that are normal in the brain for generating synchronous brain patterns are actually hijacked in hyperexcitability. So some physiological event that normally supports learning and memory in a brain that has hyperexcitability is pathologically converted to something that's now totally pathological. Another way to visualize this is that excitability is a dynamic process. So you've got these periods where there's elevated excitability around these synchronous events. And in a normal brain, that's fine. These are supporting some aspect of cognition and is well above the seizure threshold. But in a brain that's got hyperexcitability, the seizure threshold by definition has been pushed down, and these synchronous events are now converted into something that's epileptiform. So what we like about this is that it's a testable hypothesis, so we can really study the mechanisms of it, but it's also very, very simple. It's unlikely to explain, every hyperexcitable condition in the brain, but at least we can, test this across an array of different conditions and see if there's some general principles for how epileptiform activity emerges in a brain with hyperexcitability. So just to orient everyone to what synchronous activity looks like in a normal brain, it's long been known that there's generally 3 sort of brain states that are characterized by different patterns on the EEG. And it was Case Vanderwolf who really showed that this is related to behavior. So, for example, when an animal is engaged in locomotion or rapid eye movement sleep, you see these very prominent theta oscillations. And then when the animal stops moving and is still in a more aroused state, you see this very small irregular activity, so no discernible pattern, and the amplitude of the EEG is very muted. And then if the animal transitions to something that's in a more restful state, or like consuming food or non-REM sleep, this is where you see the highest amount of synchronous brain activity. So there's these very large voltage fluctuations in the brain. So when we're thinking about hyperexcitability, that's typically the brain state that we're most interested in. And so we can break that down a little further. So if we record from different locations within the hippocampus, we can separate some of these large amplitude events into different types. And so, for example, if we record up in the CA1, the output channel of the hippocampus, these are associated with this brain pattern called a sharp wave ripple. So there's these really fast oscillations that occurred around 150 hertz and looks something like this. And if you record down here in the dentate gyrus, you see these very large spike looking patterns. These are called dentate spikes. And some of these are up to 4 millivolts in a totally normal brain. So you might actually think that they look epileptiform in nature, but these are from totally control mice that don't have epilepsy. So these are really the building blocks of these sort of large irregular events. And they occur at around the same rate. So there's an equal proportion of dentate spikes and sharp wave ripples, but most of them of the dentate spikes are of this type 2 variety. And I will also show later that if we really zoom into the behavioral state of the animal, this type 2 dentate spike is a little bit different. So we see this transition to a more aroused brain state based on the pupil size. But I'll dig into that a little bit more. But what I want to say here is that by and large, our field of the hippocampus and learning and memory is largely focused on two of these sort of brain states. So theta oscillations when the animal is exploring the environment. And then when the animal transitions to this large irregular activity, our field is largely only focused on this sharp wave ripple pattern. So what we've learned about this so far is that there are specific computations that happen during these different brain states. So for example, when an animal is actively exploring an environment or navigating, there are cells in the brain called place cells that fire at specific locations in the environment. So here, the animal is just running down this linear track, and there's color-coded for different places. And certain neurons will like to fire, for example, in this blue location, some like to fire in this orange location, and collectively they tile the entire environment. So this is the way like the animal knows where they are as they navigate the environment. And you can use just this neural activity to decode where the animal is at any given time. So these are place cells. And what people noticed when the animal transitions to this rest period, where there's this really large irregular activity, is that these sharp wave ripples, this pattern recorded in CA1, are associated with replay of these place cell sequences. So for example, if the animal's resting in this kind of pinky location, you might see replay of neural activity from the beginning of the track. And so this is completely uncoupled from where the animal actually is, and is replaying these sequences of prior waking experience. So this can happen during sleep. It can happen when they're just resting or consuming food, but it's replaying neural activity that's completely uncoupled from where the animal actually is. And these are super important for consolidating memories. If you block these, I'll show this later, you don't get memory consolidation. So that's part of the reason for that. But in contrast, as I sort of alluded to before, we know virtually nothing about dentate spikes. So this equally prevalent brain pattern in the same behavioral state, this has basically been left unstudied. And so that's what I'm going to talk about today. I'm going to talk about three basic parts in this talk. So first, I just want to figure out what is the potential functional role of a dentate spike? Is it doing something like a sharp wave ripple, where it's replaying neural activity and strengthening memories? Or is it doing something completely different? I want to know what happens to these events in hyperexcitability. And you'll see from the results why that's an important question. And then finally, if there's time, I'm going to talk about how we might be able to translate some circuit interventions with some cell type specificity into a clinically relevant modality. So I will first talk a bit about the dentate spikes. So they were first identified around the same time as sharp wave ripples. So if you're recording in the hippocampus, you see this ripple activity up here in the CA1, and within the same brain state, you get these dentate spikes. And there's these two types that have different inputs that are thought to generate them based on the current source density profiles of the event. And what they found was that dentate spikes occur at around the same rate, as I mentioned before, but during the same brain state as sharp wave ripples. So primarily when the animal's resting and in non-REM sleep. But importantly, they don't occur at the same time. Very rarely do you see overlap between a sharp wave ripple and a dentate spike. So they might be involved in different mechanisms. And if you don't believe me that nobody studied this, here's just PubMed hits for studies looking at sharp wave ripples in the last few decades and dentate spikes. So it really has largely been unexplored. So I wanted to get into this, especially during COVID, when I wasn't actually collecting data, and I had all this time to analyze data. And there were some really cool recordings that you could take a look through, provided by the Allen Institute and a few other labs. So these are neuropixel recordings. They have hundreds of recording sites, and there's 6 probes going through the brain, 4 of which here are going through the hippocampus. So you can get local field potential data, the spiking activity from about 1,000 neurons from some of these recordings, and all throughout different brain regions. So the first thing I just wanted to know is, can we identify dentate spikes from these probes that are going through the hippocampus? And so here I'm showing just one example from one of the probes. I'm showing a sharp wave ripple up here in the CA1, so we can reliably identify this pattern. And then two different types of dentate spikes. So this DS1 has a pattern that looks like this and has a current sink, so it's getting inputs from a different region of the granule cell dendrites than this DS2, which has inputs coming in from a different region. Yeah, so essentially we can identify these from these large scale recordings. And just like the original paper showed, these are primarily occurring when the animal is resting. So if you look at the pupil as an index of the arousal state of the mouse, they are all associated with a reduced pupil size and a locomotor speed of basically 0. So the animal is not moving, the pupil is constricted, supporting that they're in this sort of low arousal brain state. And just like the original study, the cumulative total of dentate spikes is roughly equal to what we see with sharp wave ripples, so around 0.2 hertz, and most of them are of this type 2 variety. And you'll see why in a minute, but I'm going to largely focus on dentate spikes of this type 2. So if I say dentate spikes, I'm pretty much going to just be referring to this DS2 phenomenon. And then also, this is just quantification of, if you look at the amount of dentate spikes occurring around a sharp wave ripple, this is the percentage of dentate spikes. You see this coupling. So there are some events that do occur during a sharp wave ripple, but it's only about 6% of them. So largely, they're completely distinct phenomenon. And one thing we noticed that wasn't described in the original publication was, if you look at the brain state around the event, so up here, this is just pupil and locomotor activity at the time of the event. But if you look at, for example, movements of the face or the pupil diameter before, during, and after, we see something very different with this DS2, this type 2. So we see this rapid onset of facial movement, so they make these sort of twitches, and the pupil actually dilates immediately after the event. So they're actually transitioning to a more aroused state, specifically with this type 2 dentate spike. So we wondered whether we could simply evoke a dentate spike by arousing the animal. And sure enough, so if you play a loud tone or blow air at the mouse, we can evoke these DS2 patterns that look exactly like a spontaneous. And this was quite robust, so more than half the occasions we are able to evoke a dentate spike with a very consistent delay. So this is just to say that we can identify these dentate spikes reliably in these large-scale recordings. And the DS2, or this type 2 dentate spike, seems to be something fundamentally different. And so next, we wanted to really utilize the full power of these recordings, where we have access to the spiking activity of thousands of neurons in the brain. And so here is just spiking activity shown as a heat map. So blue would be a decrease in spiking, red would be an increase. And prior to this paper, these sharp wave ripples were regarded as the most synchronous brain pattern in the mammalian brain. And so here's actually what the spiking activity looks like. I've got brain regions here, color-coded. So purple here is the midbrain. Green is the hippocampus, where you see this large increase in activity, but this is also where we record the event and expect to see it. Yellow is the thalamus, and blue is different cortical regions. And really, so we do see a large increase in activity with sharp wave ripples, but we really only see activity increase in a few of the downstream outputs, like the lateral septum or retrosplenial cortex. And so what we were really surprised to see is that dentate spikes do something totally different. And this is why I'm interested in this type 2, because we see this very large synchronous increase in neural activity throughout the entire brain. And so we wanted to compare this head-to-head with the sharp wave ripple, since this was considered to be the most synchronous event in the brain prior to this study. And so if you just plot the difference in firing rate for a dentate spike versus a sharp wave ripple, you can see virtually all the other brain regions, other than a few hippocampal regions, are more increased during a dentate spike. And another way to look at this would be, let's just take all the spiking activity throughout the entire brain. So these are just like a raster plot of individual neurons. There's hundreds of neurons recorded here. We're going to collapse that down into a mean firing rate and just look at any time there's a large increase in firing rates across the entire brain, essentially. And you can see that some of these occur during locomotor bouts, but many of them are during this period where they're not moving. So this more low arousal brain state. And you can see there's actually quite a high rate of overlap between this DS2 phenomenon. And this is just a quantification of that data here. So if you just take all the neural activity and look at brain-wide increases in spiking activity, they are most likely to occur perfectly aligned with a DS2. So again, this is just a way to say that DS2 is doing something very different in the brain. But next, we really wanted to make sense of it and look at spatial cognition, which is something we know a lot about when the animal is actually engaged in navigation and during sharp wave ripples. But again, we don't know what actually happens during these dentate spikes. So for that, Ernie Huan, this was back when I was a postdoc in the Schultes lab. Ernie and I did these recordings where we had mice navigating a linear track, just like I kind of showed you previously, where it was sort of color-coded. Here we just have different textures along the treadmill, and they make repeated laps of this. So here's their position on a treadmill. You can see it just oscillates as they make repeated laps. And then here, this first group of cells are what we consider place cells because they reliably fire at specific places in the environment. So at the bottom end of here, we'll have some cells that are firing at the beginning of the track. And you can see that they consistently do this. And at the top here of these place cells, you have some that fire at the end of the track and consistently do this. And collectively, they tile that entire treadmill. So these place cells are the ones we think that are telling the animal where it is in space. And then what we can do is we look at all the activity that happens while the animal is immobile and split it into sharp wave ripples, which we know a lot about, and dentate spikes, which we don't know really anything about. And one thing I did was I looked at the location at where the event occurred. So I sorted the position for each event based on where the event location occurred and just bin that down to average it across all the different events we reported. And what I noticed was that we get this tiling effect that looks a lot like what happens during natural locomotion, suggesting that the place cells that are being activated are the ones where the animal currently is. So the current location of the animal is actually being encoded in the population activity. So we wanted a better way to quantify this. So what we did is we trained a decoder on all this running data and used that to infer from the neural spiking data where the animal sort of thinks it is. This is what people use to look at sharp wave ripples. Oh, we're getting a phone call.
Yeah, sorry.
Should we accept?
No.
No? Okay. So we're training a model based on the active locomotion of the mouse to decode what happens during these offline states. And so what we see for sharp wave ripples is that the decoded position is often different from the actual position of the mouse. So you get a sort of even distribution where the mind's representation of space is kind of uncoupled from the animal's current reality. But for dentate spikes, we see this over-representation of the current position of the mouse. And looking at this another way, I mentioned that during sharp wave ripples, you see replay of neural activity, often as a trajectory. So the animal, if you can think about it this way, might be imagining navigating certain parts of the treadmill. And so there's a movement of this decoded position over time. And so we see that with sharp wave ripples, and they tend to be uncoupled from this dotted line of where the animal actually is in space. But for dentate spikes, the decoded position tends to remain on that dotted line of the actual position. So we don't really see a change in the decoded position over time, suggesting that these events are really stationary and recruiting cells that are telling the animal where it is currently in its environment. So what we next wanted to do is what the sharp wave ripple field did many years ago, was try and assess what is the actual role in memory. Sure, you see these replay events, but do they play any role in supporting memory? And so what they would do is they did these closed loop experiments where you detect a sharp wave ripple. And so you can see here the start of an event, you see this oscillation. And then as it's detected, you either do an electrical stimulation or an optogenetic stimulation to shut down the neural activity and end the event early. And you can do like a control stimulation where it's delayed. So you let the neural activity play out through this event and compare memory performance on a, for example, a spatial memory task. And what all these papers found is that if you end these sharp wave ripples early, you disrupt subsequent performance on a memory task. So these events are very important for memory consolidation. But what we were really bummed to see was that during dentate spikes, the firing of the cells, by the time you've detected the peak of the event, because this peak is much shorter than a sharp wave ripple, which tends to last 50 to 100 milliseconds, dentate spike is over and done with in about 25, 30 milliseconds. And the cells have already fired. So whatever plasticity might have been going on, it's too late. So we couldn't really do this strategy. So we had to come up with another way. And so what we did was we took advantage of what I showed earlier, is that if you play a loud tone, you can actually evoke a dentate spike, and you can do this very reliably. And a loud tone is actually aversive to the animal. They don't really like it, not surprisingly. And so we can put an animal in an environment with two sides, and they're free to explore either side. But one of them is going to be paired with a very loud tone that they don't like. And if an animal's smart, it will avoid going in there. But since we know that this tone is going to evoke a dentate spike, we can deliver some optogenetic stimulation to selectively disrupt the dentate spike that's going to happen with this loud tone. And as a control, we can do an optogenetic stimulation during the actual tone exposure. So this is only 350 milliseconds, but we're going to inhibit neural activity with this optogenetic strategy, and but uncouple it from the dentate spike. So this is just proof that this works. So if we play a tone, we evoke a dentate spike. We play a tone, but now deliver light to these opsins were expressed in GABAergic cells. So we're driving the GABAergic cells just to shut down neural activity. And it works to actually shut down dentate spikes. And then we let the tissue recover. And immediately, if you play a tone, you get a dentate spike. So the intervention works quite well. And then here's what happens with a control mouse. So just a reminder, they're actually getting this stimulation to inhibit neural activity during the tone, but it's just not during a dentate spike. And you can see from this baseline to when the tone's actually paired with a location of the environment, they make this association where they avoid the side that has the loud tone. So they successfully form a memory or an associative memory. But in the mice where we disrupted the neural activity only during the dentate spike, they make no association. So this then suggests that dentate spikes are actually playing a role in acquiring new memories. So I'll just first summarize the first part of the talk. So what we showed is that there's this elevation in brain-wide firing that's quite specific for dentate spikes. Dentate spikes are associated with this transition to a more aroused brain state from generally a low arousal brain state. And then if we look at the actual population activity within the hippocampus, we see something very different from a sharp wave ripple. So during a sharp wave ripple, we see replay of prior waking patterns, and these have previously been shown to play a role in memory consolidation. But for dentate spikes, we see the activation of cells encoding the current location of the mouse. And I think this fits really nicely when we consider that this is associated with an arousal transition. So if you're a mouse in an environment in this sort of low arousal brain state and something startles you, do you really want to be replaying some random part of the environment or thinking about things in your past? Or would you rather realign your cognitive map to your current location and think about getting out of there? So that's what we think is going on with the dentate spike, is it's kind of breaking up these periods of mind wandering. And also serves a very different role in memory. So unlike playing a role in memory consolidation, we've shown evidence that at least for these evoked dentate spikes, they play a role in forming memories. Since if we block them, the animals aren't forming these associative memories. Yes, absolutely.
So with the experiment, Susie, that you're talking about, is formation. If the animal had already made the association, like that, to avoid the tone space, would that still work? Like if you now try to interrupt?
That's a great question, and a good experiment that we should do that we just have not done yet. But yeah, I'd be very curious to know.
The reason I ask is that because if it's, as you were saying, with location, like current location encoding,
yeah,
if I interrupt that, the animal doesn't know where it is, and they might.
It could just be something simple like that. Yeah, no, yeah, that'd be a great experiment to do. Yeah, and please feel free to interrupt anytime with questions, especially if there's something that needs to be clarified too. So yeah, before I move on to the next part and start talking about hyperexcitability, I just wanted to like quickly plug some work that Amanda Schott has done in my lab. So recording these patterns and separating them into the different types isn't super easy. Requires a lot of programming experience. So Amanda built this really nice tool, so a graphical user interface to go through your recordings, find all the channels where these events are happening, pick them apart, and essentially curate your data set to find robust dentate spikes. So if somebody happens to be interested in this audience, we have a tool for it. So check that out on our GitHub. But now I'm going to move into talking about what happens during hyperexcitability. So as I explained before, we get these transient increases in excitability during these synchronous events. And prior to this study, we would have considered these sharp wave ripples as candidate events where excitability is very high, and in a brain that's got a lower seizure threshold or hyperexcitable, these might be events that are now pushing the network into an epileptiform event. So now that we've done this study and seen this huge increase in activity during a dentate spike, I think we need to consider that dentate spikes might be associated with these very large elevations in excitability that might be pushing a network into a pathological state in the brain that has hyperexcitability. So to address this, I think it's first just important to point out that dentate spikes are typically bilaterally synchronous. So this is just a recording from the left and right hippocampus, and these are the different brain patterns we record. It's previously known that sharp wave ripples are synchronous between both hemispheres. We see that too. And same with dentate spikes. So if you record one on the left side, you also record one on the right side. This is just a quantification of that. About 60 to 70% of both events are synchronous. And then the amplitude's very tightly coupled. So the drive you see on one side that's driving a dentate spike is essentially the same as what we see on the other side. And if we look at those events where we didn't detect a dentate spike on one of the hemispheres, those are associated with just a lower amplitude. So they're probably, even for the events we miss, there's probably still a dentate spike there that's just not reaching threshold for detection. Yeah, we don't really know. Yeah, I think it has is very nonlinear. I don't know if it's perfectly all or none. And we do have to set a threshold for calling something a dentate spike. But for the most part, they're like large amplitude events, like with this big spike looking thing, which is generated by synchronous action potentials. So if not enough cells are firing, they probably don't generate that waveform, and that might be sort of all or none, but definitely very nonlinear. Yeah, so it's possible, like, for these asynchronous events that they're still getting a lot of excitatory drive, but just not enough cells are firing. but there's still that strong input reaching both hemispheres. So yeah, so these are bilaterally synchronous, which really helps us to understand what might be happening in epilepsy. So we have this model in the lab where we inject canic acid into one side of the hippocampus. And so this is work done by Mahad Ahmed, who's an MD PhD student still working at Stanford. And so what we do is we inject canic acid. It creates this lesion in this hippocampus. And about 2 weeks later, there's this rewiring of the circuit, and it starts producing these epileptiform discharges. So these large amplitude events, often associated with high frequency oscillations. And you can even see like the physiological activity is just completely gone. So this side is completely pathological. But on the other side, on this control side that's not injected with any sort of toxin, we see normal brain patterns. So we get sharp wave ripples and we get dentate spikes. So this allows us to assess that if we know that the drive to elicit a dentate spike or elicit a sharp wave ripple are occurring on both hemispheres, but one side's producing these pathological spikes. If we see, for example, an epileptiform spike overlap with a dentate spike, we can assume that the thing that's driving the dentate spike is the same thing that's driving the epileptiform spike. So here's just now when we break down the control side and look at the neural activity. So here's a dentate spike, and here's another dentate spike, and you can see these really nicely overlap with these large amplitude epileptiform events. And here's just a sharp wave ripple here. And it's not every dentate spike. So you can see here that there's a dentate spike that was reported, but there was no epileptiform spike. And here's just a quantification of that. So this is when you see these large interictal spikes or epileptiform discharges aligned to the peak of that, you often see a large increase in the number of dentate spikes, whereas we don't see that for sharp wave ripples, which again were previously thought to be more synchronous. And actually, when we look in this control hemisphere and compare that to an animal that doesn't have epilepsy at all, we see a reduction in the overall number of sharp wave ripples and a large increase in DS2, whereas we don't see really any change in DS1. So there's this like overall rewiring of the circuit in epilepsy, even though the lesion is quite focal. And then here's just, again, the quantification of the number of dentate spikes that are actually preceding an interictal spike or epileptiform discharge. I think it's about a third of them, and much less for sharp wave ripples. So I just wanted to quickly show some preliminary data that it's still very preliminary, but very exciting to share. So this is actually recordings from an Alzheimer's disease mouse. And we can see that before a large amplitude epileptic spike, we actually see DS2 precede it. We see this in another model in the lab where we have viral toxicity from a local injection of a virus into the hippocampus, and we see DS2 often preceding them. And a different model, so this is a genetic model of Dravet syndrome, so it's a sodium channel mutation. We see dentate spikes, we see epileptiform spikes, but they're not coupled, so they're occurring at different times. So if we're keeping track, so far we've looked at 4 models. 3 of them we see coupling, and 1 of them we don't. And like I said in the beginning, we're not going to explain, all epileptiform activity, but this hypothesis is testable, and we can look across a range of models and see what the coupling is between dentate spikes and epileptiform activity. But still, this is all very, very preliminary, and lots of work to follow up on that, but just wanted to share that. But what we were very encouraged by was the fact that the dentate spike is actually preceding the interictal spikes by about 30 to 50 milliseconds. So that allows us to potentially perform a closed loop manipulation. So the idea here is essentially we detect a dentate spike on the side opposite of the epileptiform discharges. And when we do that, we can then deliver optogenetic inhibition, kind of like what we did during that memory task to silence the neural activity. And so we can do that with a latency of about 15 milliseconds. And we have a less than 20% false positive and false negative rate. So we're accurately detecting dentate spikes. And the optogenetic inhibition works quite well. So we can actually use this to silence the spiking activity for the duration of the inhibition. Looks like we probably need to turn it on a little longer since there's still some spiking after. But it's working quite well so far. This is still a little bit preliminary. And if we do this compared to a baseline period, if we deliver closed loop stimulation, we decrease overall the number of epileptiform spikes. So at least showing that, it's more than just a relationship between dentate spikes and these epileptiform spikes. They're likely playing a very important role in driving epileptiform activity. And then, so now I'm going to move on to better understanding what is actually upstream of a dentate spike. So what are some of the inputs coming to the dentate gyrus that might be driving this activity, and see if we can manipulate those circuits. And so the one area that we study is this region of the hypothalamus. It's called the supramammillary nucleus. It's down here in the posterior and ventral hypothalamus. It sits right on top of the mammillary bodies. That's how it gets its name. And so it actually synapses specifically into the dentate gyrus onto the principal cells there. And it's been shown that this is a very, very powerful circuit. So it's involved in the coordination of spike timing. So it precisely dictates when the neurons are actually firing, which kind of fits with a potential role in eliciting dentate spikes. It's also been shown to have a role in arousal, which is, again, something we see with dentate spikes. So it checks a lot of the boxes for us. So I'm going to walk you through some of the results we have on this. One of the first things we saw with this circuit, which is a little bit of a detour from the theme of this talk, is that the cells in the supramammillary are often highly coupled to locomotion. And we found this very interesting. So here's the firing rate of a cell here in purple, and the actual locomotor speed of a rat that's navigating an environment. And you can see there's this really high correlation. And overall, if you record from many cells in this hypothalamic region, you see many are significantly positively coupled, some are negatively coupled, and then some are just not significantly coupled at all. And this looks a lot like what you see if you record from a bona fide locomotor region. So if you record in the midbrain, where locomotion circuitry is largely thought to be held, you see these cells at their firing rates are highly coupled to speed. And if you look at the distribution of the cells that you record from, it looks very similar, with some being positive, some being negative, and some not being correlated. So it looks like a locomotor region, which really is not something that is thought to be a role for the hypothalamus. But one thing that we saw that was very unique was that if we look at the timing of this correlation, so we actually had to shift the firing rate forwards and backwards in time, and found that if we shift it backwards, we get a much higher correlation, meaning that the spiking of a supramammillary neuron is predicting the future speed of the animal. So it's very upstream in this potential locomotor circuit. Whereas if you record in the midbrain, it's much closer to real time. So it's very upstream in this locomotor network. And we found that you can actually activate different populations of these cells in the supramammillary, and the control of locomotion is cell type specific. So I'm not going to go through the labeling strategy too much, but just to say that there are cells that express TAC1 or substance P, where if you optogenetically turn them on, so instead of inhibiting like what we were doing before, we're now driving activity of these cells, and the animals engage in locomotion within seconds every single time you turn on the laser. This other cell population that doesn't express TAC1, we don't really see that. So not a clear role in locomotion, and neither with our controls. And since the hypothalamus is evolutionarily conserved, and so is locomotion, we wondered, would we see the same activity in zebrafish, which diverged from a common ancestor hundreds of millions of years ago? And sure enough, if you record from, this is from the entire zebrafish brain, but there are cells in the ventral hypothalamus that express TAC1, and if you look at their activity, they increase just before the onset of locomotion, just like what we see in the mouse. So this locomotor role was super fascinating. It was really the basis of the paper, but What was equally interesting and very relevant to this talk was this population that we didn't really see a strong role in locomotion per se. So these cells that don't express TAC1, but they're very good at controlling spike timing. So this is only about 5% of the overall population of supramammillary neurons, but they're controlling the spiking activity of about 3/4 of hippocampal cells. So you can see here is just like a raster of spiking activity aligned to a 30 millisecond laser pulse. And you can see that they're increasing their spiking precisely timed with the laser stimulation. And this is just a comparison between the two supramammillary cell types. So this one is that's TAC1 negative, is very good at controlling spiking activity. So I'm actually just going to jump through this and show you that if we record from these two populations, so this is work from Dennis Kalur in the lab. So we got this fiber photometry system, which we can record from both populations of cells simultaneously with different calcium sensors. And both are correlated with locomotion. What we noticed is that these TAC1 negative cells have higher bouts of activity when the animal's not moving, which is a time when we know that these dentate spikes are occurring. So he put silicone probes into these mice to record from the hippocampus and look at dentate spikes. And this is the activity specifically lined up to a dentate spike. So for TAC1 positive, we see this little bit of an increase, but for these TAC1 negative cells, there's really large increase in activity. And this actually starts before the dentate spike, suggesting that they might be upstream in the events that actually drive a dentate spike. And so what we then did was we used optogenetics to activate these cells, specifically these TAC1 negative cells. And we noticed that we could evoke a dentate spike. And here we actually did this in a mouse that has epilepsy. So now we're recording from both hemispheres. So this is actually the control side. And then if we look on the epileptiform side, we can see that we generate these large interictal spikes. And if we just compare the success rate for when we successfully evoke a dentate spike versus not, you see this larger increase in the number of optogenetic activations. When we successfully evoke a dentate spike, we also evoke an interictal spike. So we think that this supramammillary circuit might be potentially important in both driving a dentate spike and under normal conditions, but under hyperexcitability, driving these pathological discharges. So that's sort of the overarching hypothesis that we've really started to test in the lab. And our results so far are showing that dentate spikes might play a crucial role in providing activation that generates pathological activity in a hyperexcitable brain. So in the last part of the talk, I have a little bit of time. I'm going to jump through this pretty quickly, but I wanted to show some work that we're doing to look at a potentially translatable approach to circuit modulation, since most of what I've showed is optogenetics, which we're still quite a ways away from being translatable. But I'm very encouraged by some of the work we've been doing with focused ultrasound neuromodulation. So we can get clinically very small focal regions of activation where ultrasound waves concentrate and do something to the tissue. And the main question is, what is it doing? And what we really like about ultrasound is that it's both non-invasive and can reach deep brain targets. So something like the hypothalamus is actually within reach for ultrasound, and we can do this non-invasively. So there's many applications that people are using ultrasound for example, opening the blood-brain barrier. It's now clinically approved to do targeted lesions of the thalamus, for example, in essential tremor. But what we're talking about is this lower intensity of stimulation. So we're not heating the tissue. We're not trying to create a lesion. We're just we're essentially delivering a mechanical stimulus to cause some sort of change in neural activity. And really what we want to figure out is what is it doing there? And so there's many other names for it, but we'll just call it transcranial ultrasound stimulation for now. And so work from the Shapiro lab has nicely shown that there are mechanosensitive ion channels that are expressed in the central nervous system. And what they do is they take mechanical force that's applied to the membrane, completely independent of tissue heating or bubble formation. So it's really mechanical forces acting on the membrane so that these ion channels open and conduct current and calcium and activate the cells. And it's not just one specific mechanosensitive ion channel, there's many of them, and they all play some sort of role. So for example, here is an activation with ultrasound, and you see this increase in activity within these cultured neurons, and different genes, when you knock them down for different ion channels, play varying roles. And so this is just sort of a summary of some of these different ion channels. And what's cool about this is, for example, if you take from the Allen Institute different human cortical cell types and look at the expression levels, they're variable across these different ion channels. So some are expressed in most of the cells, some only show up in a few specific clusters and in different clusters. And why this is exciting is because there's a very vast parameter space that you can explore. So you can play around with the pulse repetition frequency, the duty cycle, power compression. There's many ways you can deliver ultrasound into the brain that might do different things, especially given this heterogeneity in mechanosensitive ion channel expression. So the basic formula for this is that we know that different cells express different flavors of mechanosensitive ion channels. These ion channels probably respond to these parameters in specific ways. Therefore, we can do non-invasive but cell type specific neuromodulation if we understand what's being expressed and how the different ion channels respond to ultrasound. So we just wanted to know whether this was possible. And so Keith Murphy, while he was still a postdoc in Luis de la Chea's lab at Stanford, developed this system. So this is fiber photometry, like I showed before, where we can record calcium activity from different populations of neurons simultaneously while we're delivering ultrasound stimulation. If you're interested, you can actually get your own system now, and you don't have to build it yourself like we used to. But the point of this is we can put the calcium sensor in different cell populations, record neural activity. And what we see is that, for example, just varying one of these parameters, this is the pulse repetition frequency. So if we just deliver a single pulse, we get this activation of CAMK2 cells. So these are excitatory cells. Whereas the inhibitory cells that express parvalbumin, we don't see any change in their activity. If we now just do this at 900 hertz, so a very high pulse repetition frequency, these GABAergic cells that express PV are highly active, and actually we see a suppression of the excitatory cells. So same brain regions being stimulated, just with a few different parameters, and now we're getting cell type specific neuromodulation. And then here's just an example of how we applied this. So prior work showed that if you activate specifically this class of GABAergic cells that express PV, you can stop seizures with optogenetic means. So this is, taking transgenic mice, implanting a fiber, delivering light into the brain, so not necessarily translatable. But we did this completely non-invasively with ultrasound and saw that we could stop seizures with this optimized stimulation protocol. And here's just a summary of that data across 5 different mice. You can see when the ultrasound comes on, we stop spiking activity, and it persists for a little bit longer than the stimulation itself, similar to what we saw in the activity plots. So yeah, essentially what I showed today is we wanted to just first dig into the mechanisms that generate synchrony in the brain, and came across this event called a dentate spike, or DS2. We looked at some of the cell types and networks and behavior that occurs during these events, and what happens to hippocampal place coding. And I showed you that under normal conditions, it plays a role in realigning the cognitive map to the current location of the mouse. And it seems to be very important for encoding new memories, since if we block it, we disrupt this associated memory. And then one potential regulator of dentate spikes is the supramammillary region of the hypothalamus. And so when we activate it, we can epilepsy, we actually trigger epileptiform discharges. And so what we're currently following up on is if we can manipulate this circuit with this non-invasive approach and see if we can actually control seizure activity. And so I'll just quickly thank the people that were involved in this. So this is my lab. And I tried to thank the people that were involved as we went through the talk. And a lot of the work I showed was in my former postdoc lab with Yvonne Schultes. And big thanks to the funding. So most of the work was funded by the K99 and the Brain Initiative grant. And some of the work we're doing now is supported by Klingenstein and Cure Epilepsy. So, and thank you again for your attention. I'll take any questions.